Hey friends, welcome back to the channel. Today I'm going to elaborate all of the IMAT biology syllabus in very detail. I'm going to list down all of the topics with chapters. What are the main topics that you need to study, what you need to know about if it's reproductive system, what you need to study in reproductive system, what you need to study in cell biology, in biological molecule, what you need to study. I'm just making because a lot of students ask that IMAT syllabus, what the Italian government that publishes, is very general. So I'm just making very specific. I'm going to include all of the topics with chapters. So just let's go to screen, and this video is going to be like little bit lengthy, as we have got 47 slides, and so this video can like go up to 1 hour or 1.5 hour, but let's say tuned, it will be very valuable, I can just say. I will make just 3 master classes. First of all, this biology, and then we have got 2 down the line, that is chemistry and physics. But today we are going to discuss biology specifications, biology 1, and I have mentioned that I am a 2024 and 2025 syllabus. So let me clear that IMAT 2024 syllabus is not out yet. It is going to be like IMAT Italian Ministry publishes a syllabus in around about June, July or April. These are the 3-4 months where it publishes. But I'm saying that IMAT syllabus is almost same with like previous year. There is no big difference in all the year. Every year there is just little bit addition or subtraction, but overall syllabus remains same. If you compare 2023 syllabus with 2022 one, that is, you will find it 100% same, maybe 99% same. There will be 1% difference, so there is no big change at all. So just this is the concept. If anything like included in 2024, 2025, 2026 syllabus, you need to follow these specifications. I can just guarantee you there will be nothing out of the exam, out of the syllabus. There is like everything is included in this thing. There will be no addition or subtraction from my own side. That's all what is given in IMAT syllabus. So let's begin with our table of contents. So this is our table of contents. In table of contents we have got, I organized in this way so that it should be like easy for you to study. First of all, we have got cell biology, then we have biochemistry, bioenergetics, cell cycle, genetics, mutations and evolution, biotechnology, anatomy, physiology, animal tissues and homeostasis. I have made anatomy and physiology in red color because there are a lot of chapters included in anatomy and physiology, like the reproductive system, digestive system, that is totally, and with information with all topics. I have made separate slides. I'm going to list down each and everything. So just stay tuned, grab your hot drink, and let's just begin, or just remain with me throughout the video. Okay, now first of all, who I am. I am Sami. I paid in IMAT 2023 and got 46.2 marks. On this channel, I don't know wherever you are watching, on my YouTube channel, we explore strategies and tools that help students to become more productive in their studies. Nowadays, I'm trying to make legit videos, #flex on IMAT stuff. Or this is my Instagram handle, where you can actually ask me anything you like to. I'm totally available. I will 100% reply. And so now let's move on to our IMAT specifications, that is cell biology. In cell biology we have got these main topics: cell theory, next we have cellular structures, then we have cell membrane, prokaryotic versus eukaryotic versus. I have made separate slides like this organically. Now we are going to we are going on them step by step. Okay, first of all, you need to study in cell theory. Also, you need to study in cell biology, animal versus plant difference. I made the slide, but I did not mention here. But in prokaryotic versus eukaryotes, you need to study the difference. And I made viruses. I included viruses in cell biology because there is a reason. They should be in microorganisms, but they do not, in IMAT 2023 syllabus, there is no bacteria, like they do not mention anything about bacteria. So that's why I thought that a virus should be in cell biology. We actually study some general features of viruses, so it should be in cell biology. Now, first of all, in cell theory, what you need to study in cell theory, you need to just study main points. Introduction, like history, who discovered this, Sladen and Shawan, and then what are the main points of all living organism composed of puff cells and all that stuff. Next, there is like a statement from a scientist that is Virtue or something, I do not remember if I recall correctly. Virtue said that cell arise from pre-existing cells, so you need to remember that thing. And next, there cell theory limitation, you also need to study limitation of cell theory, and that's the main point. Then we have got cellular structures. Cellular structures are basically organelles. So now let's move on to our organelles. This is the list of 14 organelles. One is missing, glyoxysome, I did not mention here, but you need to study that too. Now, what you have to do in organelles is that you need to implement 3 things or 3 general things in organelles, on each and every organelle. First of all, the introduction, next structure, and then function. What is the structure? What is organelle actually? Then organelle's composition and made up of all that stuff, and then function. What are these organelle actually does, or what is the function? So you need to just implement these 3 structures, 3-step formula on each and every organelle. Like in nucleus, first of all, we have to know that what is nucleus. This is brain of cell, controls each and everything in cell, main functions of cell, and it is consist of nuclear membrane, then nucleolus, then chromosomes. Actually, it is basically in chromatin material, in the form of chromatin material. But in nucleolus, we have got like main things. Sorry, in nucleolus we have got DNA, that is actually chromosomes, and examiner can ask you, like many student confuses in this thing, confuses in the MCQs of chromosome. Like examiner asks what are centromeres, kinetochores, centromeres, centrioles. Sorry, centriole is separate organelle, centrosome and centromere, and all examiner can ask you, and many student confuses in this stuff. So just like write down your notes and then study them frequently. You will get to a point where you will remember anything on your fingertips. So but now this is nucleus, and then we have got mitochondria. Mitochondria is another most important organelle. It is also known as powerhouse of cell. It is also double membranous like nucleus. Mitochondria is 2 major things happen in mitochondria. First of all, Krebs cycle, and second, respiratory chain, electron transport chain. These are the 2 main things that happen in mitochondria. That's electron transport chain generates ATP, although Krebs cycle generates, but I think 3 ATPs and 12 NADH2. Yeah, I think 12 NADH2 and 3 cycles or 2 FADH2. Yeah, all that stuff you will study. I'm going to list down all this stuff in bioenergetics. Just little bit patience. I'm just kidding. Okay, now next we have endoplasmic reticulum. It is also rough endoplasmic reticulum. 2 main types: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough because it on its surface there are ribosomes. It is in smooth there are no ribosomes. Then we have got separate ribosome. Rough endoplasmic reticulum is involved in protein synthesis, like smooth endoplasmic reticulum is lipid synthesis. Then we have got Golgi apparatus. Then we have lysosomes. Lysosome is single membranous organelle, and then vacuole, cytoskeleton. In cytoskeleton, you have to know that what are microtubules, microfilament, intermediate filament. Specifically in cell cycle, we are going to study, although I will list down each and everything about cell cycle a little bit later, but like what the spindle fiber that arises in cell cycle, examiner asks what is their composition, so you have to know that what is their composition. In these types of question examiner can ask. Then we have centrioles, then we have chloroplast, cell membrane, plasma membrane. I have made separate slide on cell membrane. You will discover. I think it should not have to be chloroplast. There should be plastid, because we have got three types of plastid: chloroplast, chromoplast, and then leucoplast. The next we have nucleolus. Nucleolus is the sub part of is the component of nucleus. Nucleus consists of chromosomes and ribosomal factories. DNA and RNA synthesizes inside nucleolus. Then we have got peroxisome, and in addition to peroxisome we have got glyoxysome. So that was all about organelles. Just study from Pearson biology or from the books that you studied in your high school. You will know each and everything about them. Next we have cell membrane. Cell membrane is very, very important. That's why I have made separate slide, because there in every year there is at least one MCQs from cell membrane. There are three things that you need to know. First of all, structure, then function, and in last third thing is transport. How transport occurs, how non-polar and polar, all that stuff. Like cell membrane is semi-permeable membrane, we all know that. But in structure we have got like different models. First of all, like Robertson model, unit membrane model. I think Robertson has given unit membrane model, but the most appropriate model was of fluid mosaic model. Nicholson and Singer give that model, I think. Yeah, if I remember correctly. But that was all about structure. But in structure we have got phospholipid bilayer. Why phospholipid bilayer? This is the one layer of phospholipid, that is the second layer. It is called phospholipid bilayer. You need to know about phospholipids. What are membrane protein, receptor protein, transport protein. You need to know both all of these proteins. What is their function? What is their location? So if you know that, examiner I have asked question, I have made like this is the question mark, red question mark. There was like, I think if I remember correctly, in IMAT question paper in like past exam, examiner asked the difference between carrier protein and transport protein. So you have to know like this, what is carrier protein and transport protein. And last we have got cholesterol molecules. But first we have glycoproteins. Glycoproteins are also known as membrane proteins. If like on red blood cells there are antigens A, B, and AB, and if there is no antigen, it means that is blood group O. And if both are present, AB, you know that. But these are the antigens are basically membrane proteins, glycoproteins. And in immune system, in immunity, we are going to discuss that in a while. You will study macrophages. What is the macrophages are the cells that a type of white blood cells that engulfs foreign entity. When foreign entity or invades the foreign microorganism, invades the living body, the macrophages engulf that foreign entity and represent some parts of that microorganism or smaller or entity, foreign entity on their body, so that what are these antibodies can identify those foreign particles in future. And these foreign particles that are representing on macrophages body are known as antigen representing sites. Yeah, so that's the those are actually glycoproteins. What are glycoproteins? Carbohydrate plus proteins are known as glycoproteins. This is cholesterol molecules. So cholesterol molecules provides rigidity and phospholipid molecules provide fluidity. Yeah, I know you know, but maybe most of you do not know, but that was a basic thing. But animal versus plant cell. So animal cell versus plant cell. Cell wall. First of all, the difference is cell wall. Plant cell have cell wall and animal cell do not have cell wall. Then cell shape, then vacuole. I have told what is the vacuole, like a major big vacuole in plant cell, center vacuole, and many small vacuoles in animal cell. Centrioles. Okay, we will discuss centriole here on cytoplasmic streaming. Sorry. Okay, now next we have plastids. Plastids are actually chloroplasts are basically plastids. Plastids are in plant cell, they are absent in animal cell. Plastids are of three types. Chromoplast I have discussed earlier. Chromoplast, chloroplast, and leucoplast. So you need to remember, you need to know about the functions of all these types. Then we have lysosomes, cytoplasmic streaming. So cytoplasmic streaming occurs in animal cell, but there is no cytoplasmic streaming in plant cell. There is a reason, because cytoplasmic streaming occurs. First of all, how cytoplasmic streaming occurs? It is also known as cyclosis, the movement of organelles in cell, in cytoplasm. So cytoplasm, it is known as cytoplasmic streaming. But first of all, tell me why this occurs, how this occurs in comments. Pause this video and tell me. I know you do not know that, but cytoplasmic streaming occurs by centrioles. So in plant cell there are no centrioles, so that's why cytoplasm does not occur in plant cell. Then we have got nucleus position, many in plant, in center, in plant cell, in animal cell, in center, in plant cell, side, on side. I think, yeah, this is the on side. Then we have got cilia and flagella, plant cell have, and animal do not have. Eukaryotic versus prokaryotic difference. This is the basic difference. So the basic name is telling what are the difference between eukaryote and prokaryote. Eukaryote mean true, so it means eukaryotes have membrane bounded cell organelles, while prokaryotes do not have any membrane bounded organelles. So first of all, the difference lies in cell structure, then nucleus, genetic material. Actually, prokaryotes do not have nucleus. Then we have got genetic material lies in cytoplasm in prokaryotes, while in nucleus in eukaryotes. I have mentioned the examples. Animals and plants, fungi, protists are eukaryotic, while bacteria and archaea are prokaryotic. Then the difference lies in membrane bounded organelles. That's the basic definition. Size: ribosomes, animals 80S, in plant cell it is 70S. That's the spread per unit, and S mean the power that is during centrifugation it requires to centrifuge ribosome. In plant cell it act on 70S. In prokaryote it occurs on 70S, while in animal cell or eukaryote cell it is 80S. Okay. Then cytoplasmic streaming, then cytoskeleton. Examples: prokaryotes are bacteria and archaea. The famous bacteria is cyanobacteria. That is next we have eukaryotic, animal, plants, and fungi. You know that. So now next we have viruses. So I'm not going to define each and everything. I'm not going to read. The video will get very lengthy. Just pause this and read it. Sorry. In viruses, first you need to know about definition. What are viruses? And next we have viral structure. Viral structure is the most important. You need to know about capsid. You need to know about genome, genetic material. Either it is DNA, RNA, single stranded RNA, double stranded RNA, double stranded DNA, single stranded DNA, and envelope. Next, this is the types of viruses. It categorizes as DNA viruses, RNA viruses. I mentioned that. And then they have got retrovirus. Retrovirus is HIV and flu virus, I think. Yeah, if I remember correctly. But I think that is DNA virus. No, just check that out. But retroviruses are HIV is confirmed. Retroviruses. Then we have got bacteriophages. Viral replication, lytic and lysogenic cycles in bacteria. That's the basic. You have to study lytic and lysogenic cycle. And I think lytic and lysogenic, you need to differentiate those two cycles. Like busting occurs, the bacteria destroy. Bacteriophage actually invades bacteria. So just study from book or textbook if you have basic knowledge. I'm just not delivering lecture, I'm just explaining syllabus. Okay, now next we have viruses. In virus you need to study all these things too. Viral disease, impact of viruses on human history. Just exclude this if you do not want to. The syllabus will get too lengthy. Emerging viruses and zoonotic diseases, just forget these two things. But you should just study viral diseases. antibiotic versus antivirals. Viral diseases, antibiotics. What are antibiotics? Those acts on bacteria, while antivirals on viruses. But in viral disease, you have to study AIDS and flu. This is included in Pearson biology, but you can just see others too. Or if you have to study the cycles of bacteriophage, HIV, and influenza too. But that's you have to study. Next, we have got bacteria. I have made question mark because it was not given in syllabus. This is clearly mentioned here. This was not given in syllabus in 2023. I do not know about 2024. You can see that, but just study this because the one view is that you should study bacteria because bacteria is actually prokaryote, and the prokaryotic difference is given in IMAT syllabus. So that's the thing. You just these are all the things you have to know. Very basic things. If you want to study prokaryote, if you study prokaryote, you will definitely know all these things, like what is cell wall of this bacteria, and then we have got a cell membrane, a capsule, and slime is also included. Then we have got cytoplasm, ribosome, flagella, pili, and fimbriae. That's the extra thing you should study. It utilizes bacteria, uses it in during fusion, for conjugation, for sexual reproduction. And then no, it's actually not sexual. I think it's I think conjugation is sexual reproduction. I'm just confused. Just look at that. I have given IMAT exam for 4 months earlier. Okay, now we have got nucleoid, plasmids. Plasmids are DNA rings, central DNA rings. That is very whole biotechnology consists of these plasmids, occur due to these plasmids. Yeah, we will study in biotechnology, we'll discuss this. And then we have got endospores, mesosomes. Mesosome, what is mesosome? Just pause it and answer in comment. Mesosomes are the inward moved or inverted cell membrane, that is called mesosome. Yeah, that's the question. If in animal cell ATP synthesis occurs, energy production occurs by mitochondria, how does it occurs in prokaryotes, in eukaryotes? If it's mitochondria, in prokaryote, how does it happen? So mesosomes are involved in energy production. It produces energy currency, and mesosomes is also involved in cell replication, DNA replication, all that stuff, and transport across membrane. Okay, these are the things you need to know. Size, shape, types, growth cycle. In size, like if I say the smallest bacteria, the smallest bacteria is the size of largest virus. Have you understand? Largest virus. I think Dr. Pox virus. Pox is I think smallest. It should be like parvo. I do not know. You should check that. You should check book or Google it. But that is, I think that is 20 micrometer, and the bacteria starts from 20 micrometer. Sorry, size. And then we have got shape, then we have got types and growth cycle, log phase, lag phase, all this. But if it's included, then we have got our favorite thing, biochemistry. Yeah. First of all, in biochemistry, we have got water, then we have got biomolecules. In biomolecules, we have carbohydrate, proteins, lipid, nucleic acids, and enzymes. But water is the most important thing in the universe. Yeah, most important. Why H2O is important? You have to know hydrogen bonding and water molecule anomalous people. I have made separate slide. I have included my notes in this biochemistry so that you know, you just how I prepared all that stuff. So I just explained each and everything with my notes. We have got carbohydrates and then proteins. In carbohydrate, you need to know about composition, types, function. In protein, same as like carbohydrate, composition, types, function, in all this. And then in nucleic acid, we have got DNA, RNA, and enzyme, structure and function. Enzyme is the most important thing. But now let's go to first slide, that is carbohydrate, first biomolecule. Carbohydrate, what you need to know about carbohydrate. So I have just added my own notes that you have to know. In carbohydrate, you have to know about definition. What are carbohydrate? I have added here what are carbohydrates, hydrates of carbon, and this concept about 2:1. What is the ratio of water is the H and oxygen ratio in carbohydrate. I have mentioned this. But there is like theory that this is the compound that also contains 1:2, but it is not carb. Other stuff, sorry, this is the stuff that contains not 1:2, but it is carb, and this is the stuff that contains 2:1, but it is not carbohydrate. So that's the thing I have added here. But then we have got definition. That's the basic definition: polyhydroxy of aldoses and aldehyde and ketones. This is the glyceraldehyde, aldehydic group, 3 carbon sugar. Very basic sugar in the world. Yeah, I have made both are the simplest carbohydrates. This is the dihydroxyacetone, ketone composed of. And yeah, usually carbs consist of CHO except chitin. Chitin is consist of where we find chitin. Chitin is the cell wall of fungi. Yeah, fungi, and also the exoskeleton of arthropods exoskeleton. It consists of usually chitin is carbohydrate, and then this is he mentioned the source like for we obtain it from photosynthesis, and then we have got the blood sugar, all that stuff. Then we have classification. That's the most important thing. We have to know what are monosaccharides. I have mentioned all this stuff. Simple sugar, can't hydrolyze, all this stuff. C3 I have mentioned function. What is aldehydic is called, and what is ketonic is called. So I have named all those stuff. And then I have where we can find or the example. Now here we have got ribose, 5 carbon sugar, and also its ring form, that is between carbon number 1 and carbon number 4, and it is 5 carbon ring is called furanose. We call as furanose, C1 and C4, not C1 and C5. Always remember. Okay, now we have got why I have mentioned here why ring forms, because in aqueous medium, when we drop any sugar in aqueous medium, it forms ring structure to gain stability. Now next we have got carbohydrates, more carbohydrates, glucose, galactose, fructose. These are also monosaccharides. 6 carbon, also these are hexoses. Yeah, 6 carbon. I have mentioned where like OH group difference occurs on carbon number 3. Carbon 3, OH lies on left side of all the, but in galactose there is also on 4th number is also on left side. This is glucose. That's why glucose and galactose are epimers. We call as epimers. I have mentioned very deep information. Yeah, you are thinking, yeah, you have to study if you want to crack IMAT. This is glucopyranose. Glucose form, it is hexoses, so it forms 6 corner ring, and 6 carbon is called pyrene. And 6 carbon, it's not between it is not between C5, it is between C1 and C6. Yeah, C6. You can like see all this stuff. I have made PDFs of all of my notes. You can see that. Next we have got fructofuranose. Fructose is 5 carbon ring. This is mentioned here. Then we have got oligosaccharide, 2 units, 2 monosaccharides when combined with glycosidic bond, that is called oligodisaccharides, and glucose, maltose, glucose, fructose, sucrose, and glucose, galactose, lactose. Yeah, milk sugar. I have mentioned very deep information. Always remember, in carbohydrate we have got glycosidic bond, that is very simple covalent bond. I have like made very information, man. I'm studying. I have like studied. I just prepared from my notes. I like 70% or 60% from my notes, then from external books and all that stuff. That's why I got 46.2. Just didn't get 4 marks, more 4 marks. Yeah, but that's all. Then we have got maltose, lactose, all the information, homo, hetero, polysaccharide. So in polysaccharide we have got hetero and homo, but let's discuss polysaccharides in detail. Here we have got starch, glycogen, and cellulose chitin. Just skip it if you do not want to. This is the very information, like it starch reacts with iodine and gives blue, I2 glycogen gives red, but the rest of both of these do not give any color when combined with R2. This is the like bonding. How does bonding? In glycogen we have got glucose, but the bond is one glucose from one glucose molecule, like two molecules are joined from one glucose ring. Carbon number 1 is involved in bond formation, glycosidic bond, and from second ring, carbon number 4 is involved. So this is how C1 and C4 alpha 1,4 I have mentioned. And alpha is like if carbon OH is above, I think it's here. If here, this is, I think this is beta glucose. I do not remember correctly, but if OH would be upper and H will be lower, it would be alpha, I think. Yeah, there's a beta. This is how cellulose occurs, but do not go into deep. There is no question at all in previous exam in IMAT history, but you should, if you want to know, but for your information. Next we have got proteins. So proteins is another biological molecule. You need to know about proteins. First of all, definition. What are proteins? What is proteins? Polymers of amino acid. Then what is amino acid? Amino acid is consist of amine group and carboxylic group with hydrogen and alkyl group. Due to carboxylic group, it is called as acid, amino acid, and amine is amino. Yeah, that's what amine is NH2 group. And then we have got R. This is the variable group. Here anything can attach, like aliphatic, aromatic. It can be anything. If you have studied, you will be knowing what is aliphatic and aromatic things are. Then it could be charged, it could be basic, acid, what all these stuffs. But R is the variable group. This is the question that I think you will find in BMAT past papers and IMAT past paper. What is the variable group in amino acid? So here is the more information, like plus 2. These are proteins, cysteine, and these are sulfur containing amino acid. Sorry, and plus 2 cyanine is copper, hemoglobin is iron, and albumin contains phosphorus. All this stuff. Composition: carbon, hydrogen, oxygen, nitrogen. And we have got polymerization, translation. Polymerization leads to translation. Amino acid when amino acid combines with peptide bond. I have like written somewhere. No, I did not write, but there is basic bond that is involved is peptide bond. And always remember there will be always one less bond between with number of amino acids. If there are 3 amino acids, then there will be 2 peptide bonds. If 4, then 3 peptide bonds. If 5 amino acids, then 4 peptide. Always one less. Okay, that's all. This is the I have made like bond between cysteine and aniline. You can see that, and these are the simplest amino acid. Cysteine is the simplest. It contains alkyl group as hydrogen, and aniline contains methyl group CH3. Here we have got number and sequence. Number, sequence, and structure. Insulin and hemoglobin I just wrote down. This is what I found in Pearson biology. Insulin is more, and it is protein. It contains 51 amino acid and 49 peptide bonds, because here we have got 1 alpha chain and beta chain, 30 beta amino acid and beta chain. So it will like 29 amino acid, 29 peptide bonds, and 21 amino acid. So it will be 29 and 20, 49. Yeah. Then 574 amino acid. Here are 2 alpha chains and 2 beta chains, and 570 bonds. This is sickle cell anemia. I written down something like valine. Valine is replaced by glutamic acid, replaced by valine. Yeah, I made. Sorry. This is how RBC biconcave shape converts into sickle cell shape. Sickle cell anemia it is called. It's level of protein. This is the most important thing in protein: primary, secondary, tertiary, and quaternary. And I have mentioned like primary is linear, secondary is like a twist and twisting on coiling. Then we have got tertiary, and then we have got quaternary. I have write some examples here. Myoglobin and is tertiary, and hemoglobin is quaternary. I have like added some more information. You can study. We do not have so much time, but let's go to lipids. In what is included in what we have included in lipids. So you need to know about what is the basic bond in lipids. Ester bond. We are going to discuss it shortly. This is the example. This is the basic example of lipid. Basic definition of lipids. Then we have got the function of acyl glycerol, vexes, phospholipids. You need to know each and everything about all these things. You have to elaborate very extensively. Anything examiner can ask, but IMAT exam is international, so he's going to ask like some basic stuff. Not he will go into deep, although they go into deep, but they will be very general. They make some general stuff. But terpenoid, steroids, 4 rings plus prostaglandins, it is a local hormone. But this is all the example. Steroids are what? 6 hormone: estrogen, progesterone, and testosterone. Okay, now we have got lipids, acylglycerol. I have made some detail of acylglycerol. When one glycerol molecule combines with 3 fatty acid, it is acylglycerol. So this is how triglyceride is formed. 1 glycerol and 3 fatty acid with the removal of 3 water molecules combined with ester bonds. And then fatty acids is even number, and I have made some detail. Single bonded are saturated and double bonded are unsaturated. Next we have got, these notes are a little bit blurry. I captured these notes, and then to implement here, I just screenshot those pictures too, because I had converted them into PDF. But anyway, you can find those PDF a little bit while I will show you how you can download this. Then we have got phospholipids. This is the phospholipids that we were discussing in cell membrane. So in phospholipids we have got like phosphoric acid plus nitrogen containing base. It could be choline, it could be ethanolamine, inositol, it could be serine. These are the nitrogen bases. So 2 fatty acid with 1 glycerol and 1 phosphoric acid and with 1 nitrogenous base is called as phospholipid. And there is some for more information, like how many water molecule releases with 1 phospholipid when 1 phospholipid molecule is made. And then I have made like very deep information here. I have written down, like you need to study very hard for exam. That's how you can only crack IMAT exam. If you know tons of information, then you can easily ace IMAT exam. But anyway, so this is the structure of phospholipids. Phospholipid head is polar and tail is non-polar. This a curved line is basically known as kink, and this is double bond. So here is phospholipid. This is the phospholipid I captured, but this is the phospholipid, and this is how they join with each other, forming lipid bilayer in cell membrane. Yeah, you remind me correctly. Then we have got another biological molecule. We have got nucleic acid. This is the basic definition, and you need to know the difference between nucleotide and nucleoside. Nucleotide is nucleotide is all this, and nucleoside is this. Pentose sugar with nitrogenous base is nucleoside. Pentose sugar with nitrogenous base with phosphoric acid is nucleotide. This is all the information during the double bond, double ring structure, pyrimidines, all that stuff. Types: first we have got mono, and then we have got di, and then we have got poly. In di we have got NAD and FAD, NEAS and B3, all that stuff. These are the things that you need to cover in IMAT in biological molecules. So here I have explained messenger RNA, transfer RNA, ribosomal RNA in little bit detail, types of RNA. So these images are a little bit blurry. You can find my actual PDF notes, but here I did not mention here DNA. The first reason is that I made the ref sketch, so I do not think I should shoot that. But always remember, DNA is the most important topic, like cell membrane. Every year one question is confirmed. They will ask at least one question, so do study DNA in very detail. Do a study in DNA replication. You need to study actually DNA replication in very detail, how DNA like polymerization, then all those stages, termination and whatever. I do not remember all the stages, but that's all. This is how DNA double stranded look like. RNA is single stranded, DNA is double stranded, all that stuff. Okay, all right. Now we have got H2O. Importance of H2O. H2O is the structure of water. I have made some like figures here, truths and figures, like 20% in bone cells and 85% brain cell. If it becomes 10%, less than 10%, we become dead. we die actually. Okay, this is structure of water, and then we have got hydrogen bonding. Then it is universal solvent. What is known as universal solvent? This is like how when phospholipid is poured in water, it forms the shape of lipid bilayer. That's how cell membrane is actually formed. I written something here. Okay, then we have got specific heat of water. This is very high specific heat. Is very high. Water has very high specific heat. 4.18 joules actually. It's not gram actually, joules. Resist temperature and all that stuff. This is the heat of vaporization, and this is the point that if water does not have hydrogen bonding, water would have boiled at -80 degrees centigrade, -80. This is the most important thing about anomalous behavior. I wrote down. So when at 4 degrees centigrade water is most dense, and on 0 degrees centigrade it is the most light. Yeah, very light. That's why I floats on water surface. So I just made here. I have written, this is the basic thing. This is how you can remember easily. Like if it go if it density increases if temperature rises from 44 degrees centigrade, but if temperature decreases, then density decreases from 4 degrees centigrade. But from 0 degrees centigrade, when temperature rises, density increases. From 4 degrees centigrade, when temperature rises, density decreases. So this is like tricky part. Then ionization, raw material protection, all that stuff, how water protects. Then how, if you loving my notes, here how you can download. So this is the basic my published video on my channel. I have like explained in 4 minutes 34 seconds. Oh my god, very little time. How you can use my notes, and how you can how you can use my notes. You can just find my notes here, and also you can download for free, actually absolutely free. You can, if you do not find anything on my channel, just directly DM me on Instagram. This is my Instagram handle. So let's move on to our next chapter, that is bioenergetics. So I could have made bioenergetics. I should have I made very beautiful notes on bioenergetics. I remember each and everything right now. That covers each and everything that you need to know for IMAT exam, for any exam at high school level. It covers each and everything. So first of all, you need to know about what is ATP. What is ATP? You need to know about ATP. Then what is chlorophyll? It is actually included in bioenergy. This is like photosynthesis occurs by our chlorophyll brother. Yeah, this is our brother. It contains magnesium group, pyrrole rings, porphyrin, all that stuff. You know, this is included in structure. Then we have got types: chlorophyll A, chlorophyll B, bacterial chlorophyll, all that stuff. And then we have got photosynthesis, light reaction, dark reaction, raw materials, what are the products, all this stuff. I have explained, I have made some points for photosynthesis and respiration explicitly. Glycolysis, this is the first step of respiration. Krebs cycle, electron transport chain. And then we have got lactic acid fermentation. This is the anaerobic respiration, that is the aerobic respiration. So let's move on to our photosynthesis. In photosynthesis, we have got, you need to cover all this stuff. First of all, photosynthesis, this is basic. Like you need to know what are the raw materials for photosynthesis: sunlight, then water, then the chlorophyll. All these three things. Sunlight will fall on leaves, which contains chlorophyll, and then photosynthesis will begin. You have to know photosynthetic pigments. We have got reaction centers, and photosynthetic pigments is actually chlorophyll. But in chlorophyll, we have got reaction center and antenna complex. This is the complex information I have written down. I just think that I should make some free tutorials from my notes, but that consumes so much time and energy, man. We will decide actually. And then we have got photosynthetic pigments, light dependent reaction, light independent reaction, In light dependent reaction, photosystem 1 and photosystem 2, photosystem 1 is 700 nanometer, absorbed wavelength of 700 nanometer, and this is the 680 nanometer. Electron transport chain, chemiosmosis and ATP synthesis occur in chlorophyll B and 6, BF, chlorophyll BF, B and F. Yeah, just look at Pearson book, you will get to know. Light independent, all this stuff. Carbon fixation is the part of light independent reaction, Calvin cycle. Then we have got reduction, and then we have got regeneration, carbohydrate formation. Then we have got regeneration, carbon fixation, reduction and regeneration. Factors affecting photosynthesis, importance of photosynthesis versus cellular respiration. This is the most important. Breathing and you can just we will discuss that later. Okay, now this was photosynthesis. Then in respiration, you need to know about glycolysis, transition reaction. After glycolysis, the pyruvic acid go to addition reaction, that is transition reaction. We call it transition reaction, that occurs right before starting of Krebs cycle, citric acid cycle. Then we have got electron transport chain, energy production, chemiosmosis and ATP synthesis, all that occurs in here. Anaerobic respiration is fermentation, lactic acid and alcoholic. Aerobic respiration versus anaerobic respiration, respiratory. This was cellular respiration was aerobic respiration. Then importance of cellular respiration, energy used in cellular respiration. That is most important. How much energy? How much energy ATP generates? 7.3 kilocalories. And how much ATP is formed in one complete respiration? That's a net 36 ATP in eukaryotes and 38 ATPs in prokaryotes. Why I'm telling you, can just, when you will study, you will go through all these things. Then factors affecting cellular respiration, connection between photosynthesis and cellular respiration. So you just study respiration, complete respiration. Glycolysis, Krebs cycle, how much energy, how many NADH2 occurs, what is consuming, what is forming, how alpha ketoglutarate converts into succinic acid, succinyl coenzyme, succinate into fumarate, then malate, then what is citric? What is the 4-carbon compound? The first compound, citric acid? No. Yeah, I think it's citric acid. Just look at my notes. Okay, then we have got cell cycle, most important, and this is also the most important chapter. In cell cycle, you have to know about S phase. Two main things you need to know in cell cycle: mitosis and meiosis. These two are the things. I have made very basic and good notes on this. S phase, main points, and division phase is the M phase, that is 2 main points. But what are these 2 main points? Mitosis and meiosis. You need to know stages, meiosis and stages in what are also. You have to know chromosomes for centrioles. Why I mentioned centriole? This is not. This should not be. It should be centrosome and centromere or kinetochore, all that stuff. Okay, now we have got interphase. Interphase have got S phase, G1, G2, G2 and G0. Interphase is also known as S phase. Sometimes we call it. Just you have to know this is you do not have to know all this stuff, but this is the for formality, for just if you are following some books, and if you want to say that I am clear in this chapter, then just go through, make sure that you have gone through all this list. S phase, G1 phase, G2, G0 phase. This is how cell cycle occurs. G1, S phase, then G2, then M phase, then G1 again, then G0. If a cell does not want to go to S phase, then it converts into G0 phase. It goes into G0 phase, and you have to know that 90% of the time cell remains in interphase. Only 10% of the time it is in division phase, mitosis or meiosis, whatever what is going to happen. But always remember, if we have got 24 hours, 2-3 hours, the cell will remain in interphase, where DNA replication occurs, cell organelles grow, all the stuff, cell organelles duplicate, all the stuff. Okay, then we have got G2. You should know all the stuff. So now let's move on to division phase. We have got 2 main stages, mitosis and meiosis. What is mitosis? Mitosis interface, G1, all this stuff will occur in a sequence, G1, then S phase, then G2 phase. Then we have got mitosis. Prophase, like in prophase, what happens in metaphase? I have like, this is the cell cycle. This is what it is. This is prophase, this is metaphase, cell aligning at metaphase plate. This is actually metaphase plate. This is S phase, interphase. This is my prophase, metaphase, anaphase, telophase, and then cytokinesis. Yeah, so division cytoplasm. This is the division of cytoplasm, division occurs. So you have to know all these stages in mitosis, and you have to know how many chromosomes or how many centromeres are in prophase, metaphase, 4 and 2 and all that stuff. You have to know all that stuff. Then we have got meiosis. Meiosis is similar to mitosis. Actually, the meiosis 2 is same as, but in meiosis 1, instead of one chromosome, homologous chromosome lines at metaphase plate, and when meiosis 1 completes, cell chromosome number becomes half. So this is the basic difference, and meiosis is sexual reproduction and mitosis is asexual reproduction. If you are enjoying this video so far, I'm just tired, I'm just breathing. If you are enjoying this video, then do subscribe to my channel, Sami Kamar. We have got like so far 4508 subscribers. I do not know why I mentioned here. Just give it a subscription so that it's totally free. Just we are going to drop very cool content like about chemistry and physics and some webinars too. So do subscribe it. The cool stuff is coming. Do join. Okay, now next we have got, so this is the reminder. There are 2 more classes about chemistry and physics, same as biology, chemistry and physics. Okay, that will be very valuable. I'm going to list down each and everything. So now we have got next molecular genetics. So I should have like first main thing genetics, then molecular genetics, and then cellular, or I do not think what I mentioned there. But first of all, we have got molecular genetics. In this thing, you have to know what is gene, structure and regulation. What is the structure of gene? Like we have got promoter region, regulator region, introns, exon. What is the difference between prokaryotic gene and eukaryotic gene? So this is the little bit thing that you need to know. Allele. The genes have 2 forms, that is known as allele, DNA, duplication and structure. Replication, actually, I made duplication. It is actually replication. You need to know the enzymes involved, DNA, helicase. Actually, the main important, the enzyme that they have asked in previous IMAT exam, DNA ligase, that is used to make that act as glue to bond Okazaki fragments in DNA replication in last stage. Then you need to know genetic code. I bet you one question from genetic code, confirm codons. Their codons is 3 nitrogenous bases. What are these? Nitrogen purines. Yeah, these are nitrogenous bases. What are the 3 adenine, guanine, uracil, and this include the 4th one. Okay, then thymine. Genetic code. Then you have to know about RNA. Similar thing, transcription and translation. This is the central dogma of life. Transcription is from DNA information into RNA information, and then from RNA information into protein is central dogma. DNA to RNA is transcription. RNA to proteins is translation. Yeah, you got me right. Then you need to know about what is the difference between prokaryotic DNA, circular and single stranded, all that stuff. Then we have got genetics. Main thing, Mendelian genetics. What is the monohybrid cross? Who was Mendel? How Mendel performed each and everything. When Mendel published papers, Pearson Biology has explained very like made crosses, how monohybrid cross, dihybrid cross in very in very informative way. That's lengthy book, you should like follow that. But monohybrid cars, dihybrid car, how what is law of segregation, law of independent assortment, so you need to cover all this stuff in Mendelian genetics. Mendelian genetics is not short, it has got like types of, I do not have made slides, but I do not know why. But in segregation you have to know what is the, like, you can draw crosses. If gametes are given, you can draw crosses. How you will obtain F1 generation, what is the what is it called, yeah, you can like conclude that what is genotypic ratio, what is phenotypic ratio, yeah, these are all these things. And you can draw Punnett square. This is the main thing. I think I have made this is the main thing. You need to know in genetics that you can draw crosses. Pedigree is the most important thing. 1 or 2 questions are absolutely confirmed from genetics and from majorly from pedigree analysis. Pedigree analysis is very easy. Many students makes it that it is very hard. Although it is, I say that it is hard, but you need to learn like a rule. I have like made, I think in a rough work, I made like very small, what we call as short tricks to remember if dominant occur. That's actually pedigree analysis practice, basically practice. You have to practice very extensively to master what pedigree. But now we have got chromosome, then we have got chromosome maps, how to draw. Morgan has given the concept of chromosome maps, like you can draw. I have made very beautiful notes on this. Then we have got chromosome set. This chromosome maps is very easy to draw. Then we have got chromosome set. You have to know what is 2n haploid and diploid, and then we have got to know that what is trisomy, and we will discuss this in mutation actually. Okay, now we have got, then in genetics we have got reproduction. Hey, the reproduction should not be here, but anyway I have made it. It should be in anatomy and physiology. But anyway, then we have got inheritance. I think I will discuss these things in inheritance again, but now let's move on to reproduction. So in reproduction we have got to know 2 main things: reproduction in man and reproduction in female. Male reproductive system, and actually reproduction occurs between male and females, but now you need to know about male reproductive system and then female reproductive system. So in male reproduction you need to know what are testes, what is scrotum and epididymis, the difference, all these vessels, and then we have prostate gland, and the most important thing is spermatogenesis and hormones that control this thing. So you have to know these glands: seminal vesicles, prostate glands. I should have like made a list like glands and then prostate, pulpourethral gland. But anyway, so these are the glands. You have to know the functions. I have included each, and I made whole male reproduction on 1 page of my notes. That was like small content on a full page. And urethra, penis, scrotum, spermatogenesis and hormones is the most important. Examiner can ask, primary spermatocyte is diploid or haploid. He can ask. And if we have got 10 kg chromosome content in secondary primary spermatocyte, how much content we will get in sperm? So that's basically 5 kilogram actually. This was example. I have included this on my notes, I think. Yeah. Then hormones, and this is the male reproductive system. Okay, then we have got female reproductive system. Female reproductive system, this is the like most complex system. It is consist of ovaries, then fallopian tubes, fallopian tubules or oviducts. Fallopian fertilization occurs in fallopian tubules. Ampulla is the part where it occurs, proximal part of fallopian tubes. Then uterus is the part. It is pear-shaped where implantation of zygote occurs until birth. Then we have cervix, then vagina, then menstrual cycle. Menstrual cycle is the most important. That's why I have made like hormones. You need to, one question can examiner can ask from menstrual cycle. There are two things, like you need to know about ovulation and hormones. And one more thing, oogenesis. You have to know about in the female reproductive system how oogenesis occurs. Oogenesis, all the stuff. Primary oocytes, secondary oocytes. Then if fertilization does not occur, then secondary oocyte will die. If fertilization occurs, secondary oocyte will convert into egg and will help in convert into egg and fertilize, and immediately fertilizes with sperm and convert into zygote. Then after 9 months it will be a baby. Then we have got estrogen, progesterone. You have to know these hormones. So examiner will ask when estrogen will high, when progesterone will be high, what is the role of follicle stimulating hormone, luteinizing hormone, and prolactin. We have got I think I do not mention these two systems in physiology. First of all is nervous coordination and chemical coordination. Yeah, okay. In nervous coordination, neuron, brain, and spinal cord. So that was the thing. But in chemical coordination, you have to master all of the hormones. Like master gland is pituitary gland, anterior pituitary gland. It secretes hormones, all hormones, major hormones, and controls the function of other other glands too. But pituitary gland is a master gland. It secretes like hormones, TSH, FSH, luteinizing hormone, then prolactin, oxytocin. Oxytocin is secreted by posterior pituitary gland, and antidiuretic hormone. And then acetyl, what are these hormones? All those hormones are included in chemical coordination, nervous coordination. Okay, then next we have got inheritance. So in inheritance we have got Greger-Mendel's law. Genetic terminology are the basic things. So what are these? Alleles, genes, Punnett square, and upper degrees, all these stuff. But degree analysis is something else. But we have got types of dominance. Or first of all, the complete dominance is in Greger-Mendel's law, but incomplete dominance, codominance, and overdominance are exceptional, are the things that violates Greger-Mendel's law. If you follow my notes, you will understand what I'm saying. But actually, I'm not saying you follow my note. Make your own notes and just revise from them. I just saying I made my notes, I studied from my notes, and that's why I know each and everything about from my notes. Okay, that's all thing. Overdominance, but then we have got multiple alleles. That's the codominance. ABO blood group you have to cover in multiple alleles. This is the phenomena of multiple alleles. ABO blood group, RH group. In 10 we have got polygenic inheritance, and 11 is sex-linked inheritance. Sex-linked inheritance. This is the most important. Like if it is X-linked recessive trait, how it will move in pedigree. If it is X-linked dominant, it is autosomal recessive, autosomal dominant, how it will go in throughout the generation. So you need to just remember this thing. And then we have got pedigree analysis. Pedigree analysis is the most important thing. Chromosomal basis of inheritance. And then we have got homogenetic disorder like hemophilia, color blindness, sorry. And next we have got baldness. I do not remember what is the name of that disease, but that is something. Then we have got mutations. Mutation, how many slides are left? Okay, a little bit. Mutations. Then in mutations, you have to know the types. What are the main types of mutations? Then we have got the somatic and germ mutation. There was question from like germ, from mutation. Yeah, from germ cells mutation, that if mutation occur in which cell will go in like pedigree or will follow in zygote or in child. That was, I think, if I remember correctly, that was the question. It occurs in sex cells, or its somatic addition, that is occur in normal body cells. It cannot proceed, it cannot be inherited by childs. So addition, substitution, inversion, all these are the types. Evolution is the main thing. In evolution, we have got first of all theories. What are the theories? Special creation, Aristotle gave it. Yeah, I think. Then uniformitarianism, catastrophism, all these theories you need to heckle and all these scientists. You need to just go through this. They will not going to ask you. Actually, they will going to ask you from Lamarckism and Darwinism. Darwin and Lamarck's theory. What was Lamarck's theory? He said that use and disuse of organs will not, if organ is used, it will be in next generation, but if it is not used, it will be frequently in while evolution, it will fade away from body or anything, just like that. I cannot be able to explain it. My English is a little bit weak, okay, but anyway, in Darwinism is the most important. This theory is very, very important. Why Darwinism is important? Darwin was naturalist. He got into like yachts from around the globe for making map for military navy, but he gave it the most important, natural selection. One question in like every 2 years or after every 3 years, there is at least one question from Darwinism. What is it? Says how natural selection occurs. It will give you some example and say what was the main principle, disused or use of organ, inheritance of acquired traits. That was the point of Lamarck. But they like give some 5 options of all these, and in last the true option will be like natural selection. And with Darwinism, we have got Alfred Wallace theory. He made some points, and then we have got some evidences of evolution. How we identify evolution? By biochemistry, anatomy. In anatomy, we have got homologous structures and analogous structures in vestigial organs like tailbones in humans, and sheathing membrane in eyes, and nipples in males, and like all this stuff. And embryology, like all, it says that Haeckel says that I do not remember more, omnis cellula e cellula, I think this was his phrase, like all stages, like all the stages are same for each creation, for each living organism on this earth. He said that there is like exception, something we do not know it's true, or it's scientifically approved. That embryology, like first of all, it goes to like blastula, glastula, all these stages are same for all mammals or maybe fishes, all this stuff. But next, move on to biotechnology. Biotechnology is most important. Recombinant DNA, it's just basic. You need to know about recombinant DNA technology. How does it occurs? Applications. PCR, polymerization chain reaction is important, and vectors. The vectors are plasmids are important. You need to remember some plasmids, or you actually need to remember the basic points of recombinant DNA technology. But let's move on to biotechnology. What you need to study in biotechnology? First of all, what is biotechnology? Historical development. Just exclude this. The main important topic is recombinant DNA technology, genetic engineering techniques, cloning and its applications, polymerase chain reaction, DNA sequencing, gene therapy. Gene therapy is also important. DNA sequencing is also important. These are the points. These are the steps of recombinant DNA technology. We have agriculture, biotechnology, history. These are the applications. If you want to study, do study them. Next we have got anatomy and physiology. This is going to cover rest of our majority of our biology syllabus. I am at biology syllabus. First of all, we have got cellular structures and function. For what? I do not give it the name. Okay, sorry, this is anatomy and physiology. So first of all, you have to know cellular structures and function. We have discussed this in very detail. Cellular membranes, cellular cell structures and types, all the stuff. But cell organelles, cell membranes, all this stuff. But now we have got tissues. Tissues is the most confusing topic for me. At my time was most important confusing topic for me. Like, you need to know what is epithelial, connective tissues, what are connective epithelial tissues, muscle, nervous, all this. We know that cardiac muscles occur in cardiac tissue will be found in heart, but that's easy. But I was confused in squamous epithelium and cuboidal epithelium, where there's like in lungs we have got like squamous. Now I am also confused. Cuboidal in like bronchioles or bronchials, all stuff in respiratory system, but that's not I mentioned. You have to study respiratory system too, tissues. And then we have got integumentary system. Integumentary system is involved in immunity. Skin structure is the first line of defense in immune system. Then we have to know hair, nails are all these are for not immunity. But here is the associated gland. Glands are the most important thing you have to know. You have to cover chemical coordination, pituitary gland. Then we have got thyroid gland, parathyroid gland. Then we have got sex glands, sex hormones actually, from hormones released from testes and ovaries. Then we have got skeletal system. Skeletal system is also important. Bone structure, type and function. What are bones? The difference between bones and cartilage. All this stuff. Joints are the most important thing and most important topic, and their classifications and diseases. Like for example, what is arthritis, sprain. Sprain is a disease. Spondylosis. I think this is also disc slip. These are the diseases. Just go through thoroughly. Just go through them thoroughly. Do not go in depth. But then we have got muscular system. Types of muscles: skeletal, smooth, cardiac muscle. This is also very important. Muscle structure and function. This is very important. Muscle structure and myoglobin. Myoglobin is blood oxygen transporting protein. Here we have got myosin and actin, tropomyosin, troponin. And you have to know all this stuff. What are A bands, I bands, what are anisotropic, isotropic A band, or anisotropic I band. And then you have to know that what is sarcoplasmic reticulum. Smooth endoplasmic reticulum in muscles is known as sarcoplasmic reticulum. It releases calcium ions that helps in muscle contraction and relaxation. Muscle contraction is by calcium ions. Muscle relaxation is by ATP. When ATP covers the head of myosin, it is muscle get relaxed. When it gets away, actin combines with myosin and contraction occurs. So then we have got our endocrine system. Actually I have made why I was thinking that I did not mention. Okay, nervous system and endocrine system. Nervous system, you have to know about nervous tissues, neurons. What are neurons? Bipolar neurons, types, motor neurons, and associated neurons, sensory neurons. Then the central and peripheral nervous system. These are the most important things you need to cover in nervous system. Brain cells, brain, spinal cord. In brain we have to first of all know the function of each and everything. In 2023 there was one function from brain. It was actually not brain. The correct option was gland, but not brain. But the question was from these two chapters. What is coordination? Nervous coordination and chemical coordination from these. So nervous system and endocrine systems are also important. So you have to know the functions of each and every brain part. What does cerebrum does, what are the functions controlled by cerebrum, what is brainstem, what is the function of hippocampus, what is the main function, what are the main functions of hypothalamus. I made the list of functions. You can just DM me on Instagram. I will give you for free, absolutely free, no money at all. I will not charge you for anything at all. That's my weird. I'm not flexing, but that's it should be like this. I'm making this to be quality. Now we have got endocrine system, all endocrine glands and hormones, regulation of what are the functions. Then we have got cardiovascular system. This is also very important. Heart and anatomy and function. What there are two main types of circulation. First is systemic circulation. Then the second one is the artery or vein that transports blood from heart to lungs. It is, I do not remember main. That is the what was that, my brain? What was that? I forgot that. Yeah. Okay, just check that whatever their name is, that is the second circulation, from heart to lung, and then lung to heart. This is one circulation, and then systemic circulation is all over the body. So endocrine system, circular and cardiovascular system. In heart we have got blood vessels and blood circulation. I have explained. In heart anatomy you need to know about ventricles, auricles, how SA nodes, AV nodes, how contraction occurs, how blood comes from body to heart to body, all this stuff, which nerve or which artery conveys blood to which part, all this stuff. Okay, then we have got lymphatic system. Lymphatic system is very, very short and very, very simple. Lymphatic organs and tissues. Lymph is actually plasma. Immune system. In lymphatic system we have got immune system function. You have to know first line of defense: skin, hair, nails, upper layer, and then second layer we have got macrophages, neutrophils, white blood cells, all this stuff, and third line of defense: interferons, antibodies. So you need to go through all these things. Then we have got respiratory system, structure and function of the respiratory organs. Lung, respiratory surfaces and lungs, then bronchiole, bronchi, trachea, nose, nostrils, and then breathing, contraction and relaxation of smooth intercostal muscles, phrenic nerves and intercostal nerves and diaphragm, all that stuff you have to cover in respiratory. How gas exchange occurs, respiratory respiration occurs. Digestive system. In digestive system, structure and function of the digestive organs, nutrients absorption and metabolism. Digestive system, from mouth, teeth, tongue to anus. You have to cover all this stuff. Here will be stomach, liver, pancreas, then small intestine. Where does absorption of or digestion of lipids occur, fatty acids occur? Where does what I'm saying? What is the main function of liver, gallbladder, bile, pancreatic juice? What are the enzyme, pH, and then absorption in small intestine, and then villi, microvilli, rugae. What is all these stuff? What are these things? And then large intestine, and what in small intestine? What is the function of duodenum, and then jejunum, ileum? What are the hormones that release in secretin, and then cyto. I forgot it, but anyway, so all this stuff you have to remember all this stuff until anus. And now next we have got a urinary system. Urinary system is actually kidney, nephron, pyramids. Then we have got ureters. We have got what is micturition, emptying of what is written here, ureter and urinary bag. Emptying of urinary bag is called, I forget the name, but kidney, anatomy and function, urinary tract and urine formation. You have to cover this. In this thing, you will have to also know the functions of like basic topic is nephron. What are proximal convoluted tubules, Bowman capsule, glomerulus, how water comes in, what is the role of efferent arterioles A and E. You have to know all this stuff. E for exit and A for I have made very short things. I just hope that, man, I can just explain all of my notes with one lecture. I know if I sit for explanation, it will take ages, and I cannot make courses. I cannot. But anyway, so we have got a urinary system, then we have got reproductive system. I have already explained what male reproductive system and female reproductive system. And the most important thing in reproductive system is menstrual cycle. When ovulation occurs. When will ovulation occur? Ovulation will occur when LH concentration will rise. A surge of LH will cause ovulation. Yeah. Evolution, ovulation, all these things, the phrases are same. But anyway, then homeostasis and regulation. How does body maintains internal environments with outer environments? It includes like water, salts, and all these. You have to maintaining internal balance. You need to cover all these things in homeostasis. There was one general question from all of the homeostasis in BMAT or I think IMAT past paper. Then we have got human development. This is not was included in 2023, but it was in 2022. So human development, embryonic and fetal stages, I did not study. You do not have to study. Like first week, 2 weeks. I just say that in my high school I did not also study this chapter at all. It was my choice, and hopefully there was it, and I was like damn sure. I was lucky that it did not appear in my final papers. And then we have got growth and aging process, and that was all. Thank you for watching, and this is my Instagram handle, and rate down if you have come so far. But if you have enjoyed this video, then give it a like, and rate video how much you have got valuable or it was not valuable. And down to download notes, the video will be in video description, and all the resources and IMAT 2023 syllabus will be in video description. And do subscribe this channel, and if you are new to IMAT, then you will love to check out this over here video and about my Notion setup for complete IMAT preparation. That includes note taking system, how to take notes, how to make questions, how to write down notes from whatever the lectures you are watching. So just give it a shot. You will love it. Thank you for watching. See you in next video of these master classes, either chemistry or physics. Thank you for watching. See you in next video. Till bye bye. Always check the playlist on my channel, so you will find videos over there. So that was from my side. If you love it, then just support it and give it a sub to channel. So bye bye.